Good morning. We'll start in a couple of minutes. Okay. So, in our previous class, we have discussed about the various types of microscope, the simple microscope, the compound microscope. Which of these two types of microscopes has a higher magnification?
Compound microscope.
Compound
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 microscope.
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 The compound microscope.
In a
Compound
in
microscope.
a
Compound microscope.
in a compound microscope, how many lenses are present?
Two.
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 Two lenses.
The two.
And how many mirrors may be present?
One.
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 One mirror.
May or may not be present, but if it is present, one mirror is sufficient. What is the nature of the mirror?
Concave. Concave.
Concave.
Concave mirror. Okay. Now, the source of the light for the simple microscope and compound microscope is The which light?
Visible light.
Visible light.
Visible.
Now, these two types of microscope, simple and compound microscope, we are able to observe the microorganisms. Okay, the scale is in micrometers. But if it is required that we want to observe some viruses or some structures which are there inside the cell, then we need much higher magnification. To get such a magnification, the white light or the visible light is not sufficient. Then we move on to using the electron microscope. The electron microscope has much higher magnification. Here we need to also understand that just if we are keep on magnifying anything, it is not sufficient. For example, using your mobile phone, you may take a photo. After you have captured a picture, using your fingers, you are able to zoom inside, into the picture. You are able to zoom. Beyond a certain zoom, what happens is you start observing that the picture is having dots. Okay? Lot of dots will be there. These are the pixels. So there is a limit how much you can magnify. Okay. So if you keep on magnifying, you will reach a certain stage where you will be able to see the individual dots. Okay. You will not be able to see any more details. Therefore, it is not sufficient to simply magnify. You need greater, what we say,
Focus.
resolution. Resolution. We need greater resolution. When we say resolution, that means the ability to see two very closely placed points as separate. Okay. The electron microscope has an excellent resolution. Okay. We are able to go much more into the details, okay, using the electron microscope. This will be in nanometer scale. The source of the light for the electron microscope is the electron beam. Electron beam. So in the top, so what you are seeing is a original setup in which there is a huge cylinder almost up to the height of the room, and in the top region there is what we say the electron beam generator. Here the electron beam, which is also known as the cathode ray, which you will study in chemistry, is generated. This beam will move downwards. On the either side there are magnets, powerful magnets. These magnets will focus this beam. Just like the compound microscope and simple microscope, where we are using the glass lens to focus the light, similarly in an electron microscope we will be using the magnets, powerful magnets to focus the electron beam. Now this electron beam is going down, and here in between we have the specimen holder. Here we keep our whatever sample we want to observe. So this electron beam will pass through the sample. If the sample is having anything structures, any structures, less amount of light will pass through it. Okay. If it is more of a transparent kind of nature, more light will pass through. All of these you will be able to observe here at the bottom of the microscope, which is the viewing chamber. Okay. Here in the bottom we can view that directly also on a whatever the rays are following on a photographic film kind of thing, and the same the digital display will be available on the computer. Okay, so we can adjust fine focus. Okay, and we can see in live how the different structures of the cell are there. Yes, there is one major difference. The electron beam is very powerful. Is the electron beam high energy, or the normal visible light is more energy?
Sir, electron.
Electron beam.
Sir,
Electron.
electron.
An electron beam.
Electron beam is very high energy. Okay, so because the electron beam is very high energy, if you keep any living cell, the living cell will die. Okay, so the electron microscope, we cannot study the living cells. We need to fix them. We need to kill them first using formaldehyde and all. So we fix it. Okay, that is a process that is called fixation. So we'll fix the sample and then only we can observe. But in the compound microscope and simple microscope, you can observe the living cells. Okay, no problem. So if we compare all the three based upon the different criteria, okay, the simple microscope, compound microscope and electron microscope, in a table form you can compare who invented it, what was the different design, what is the light source, and how much magnification we are getting. Okay, so this will summarize. The magnification that you are getting from simple microscope is maximum 200x. In a compound microscope, 2000 times you can magnify it, and in an electron microscope, almost 1 million times you can magnify it. Next, do you have any doubts with this microscope part? Shall we move on?
No.
Okay.
No, sir.
Now we are moving to the next part of the chapter, which is called the cell theory. Cell theory. So what is this cell theory? Here there are the two scientists, Matthias Schleiden and Theodor Schwann. Two scientists. One is a botanist, other is a zoologist. Independently, both of them, what they observed, that when you take the sections of the plant and look under the microscope, there are small chambers, cell like structures. Okay, just like the Robert Hooke he told. So the smallest unit of the structure of all living organisms is the cell. The other point is the unit of function of all living organisms is the cell. The first 2 points were proposed by both Schleiden and Schwann. Therefore, in the cell theory, all living organisms we refer to all living organisms, plants, animals, whatever it may be. Okay. So what is a cell? The cell theory, what it states? It states that the unit of the smallest unit of the structure of our living organisms is the cell, and the cell is the unit of the function of all living organisms. Okay. Rudolf Virchow, he proposed that all cells arise from the pre-existing cells. So what will it mean? All cells arise from pre-existing cells.
Sir, omnicellular is cellular from this word it came.
Sorry. Omnicellular. Okay, I'm not sure about what it is. But in normal terms, can you say something like, all cells
Yes,
arise?
sir. That all the cells which means the cells do not have a definite period when they will die. So the when the cells die, the younger cells grow up, and that younger cells divide into two parts, and then they form that part. So this is called the pre-existing chain.
Okay, okay, maybe we'll
And
try.
then
Maybe I think you were trying to arrive that. Okay, we'll try to put it in a simpler terms. The cells arise from pre-existing cells. So suppose you are looking at the water sample under a microscope, some kind of pond water you are using. Then you saw that there are some cells. This you are thinking is something of a new type of cell which no one has seen earlier. Okay, no one saw earlier that type of cell. Okay, some strange looking cell with two horns maybe. So can we say that this cell has come by magic?
no, sir.
No, sir.
What Rudolf Virchow has told is that every
All
cell would have come from a pre-existing cell.
by
Maybe
multiply.
yes. Now, this is the only statement he has given, that all cells arise from pre-existing cells. So what is the implication of that statement? The implication is that the cells will divide. There will be cell division. Okay. So if we are seeing any cell, that means it would have come from an earlier existing cell, and therefore the concept of a cell division arises. Whenever we are trying to define life, then we use different parameters. Okay, we use different parameters. One of that is reproduction. Can you say reproduction is the fundamental property of life? Anything
Yes, sir.
anything that is alive will die. Okay, so earlier it is happy, now it is dying. Okay, so that is fact. That is fact. Now if just there is a cell and it died, then where is the life? There is no life. Okay, there has to be a between chain where there is a multiplication of that organism, there is a reproduction, making the copies. Okay, that has to be there, otherwise that particular species becomes extinct. Okay, so the cell theory has three very important points. Number one, that cells are the smallest unit of the structure of all living organisms. Cell is the unit of function of all living organisms, and that all cells will arise from the pre-existing cells. Okay, this is the very simple but a fundamental theory. Okay, cell theory. And what it implies: structural unit, functional unit, that the cells will die and they will be replaced, and most of the life is starting from a single cell, all these things. Anything that you would like to ask in cell theory? You have any doubt in this?
Sir,
Yes.
sir, it means pre-existing cells are the unit of life.
there is a pre-existing cell cannot be called unit of life. That is only we are telling a specific what is there before. That is nothing now.
Yes, sir.
Okay, so let us move on to the next topic. That is the how many cells. Okay, how many? The living organisms, they can be either single celled, the living organisms can be single celled or they may be many celled. Okay, if there are the one celled, they are known as the unicellular organisms. Many celled, they are called the multicellular organisms. Some of the unicellular organisms include the bacteria, amoeba, euglena, paramecium, chlorella, yeast. All of these we can look under a normal compound microscope. Okay, and you can see the diversity of the structure of the cell. They are all only one cell, but you can look at how diverse they are looking. But the multicellular organisms, under that one we can see that they may be few celled. Okay, some of them may not be like too many cells also. Okay, like a Spirogyra, Volvox, tardigrades. Okay, now here this is Spirogyra, Volvox, and all these single cell organisms here. All of this we can easily observe in the pond water. Okay, or if there is a puddle of water, from that also you can take. So if there is a sunlight, then you can see that these green color ones are there, no, microorganisms with a green color. They will do photosynthesis and they will survive. These tardigrades are looking like some kind of imaginary outer space organism. Okay, these are very small animals, very small, but they are able to survive very high radiation and all. Okay, that's also, but they are also made up of very few cells. Okay, so under multicellular we are seeing at two categories, a few cells and some are made up of too many cells. Okay, for example, plants, trees, humans, all these, we are all made up of too many cells, too many types of cells, again tissues, organs and so on. Okay, so this is a overall view that when we are looking at cells, each cell can be independent like the unicellular organism, or the individual cells may cooperate with each other, collaborate with each other, work with each other as one unit to make one organism a higher organism. And sometimes the complexity may be very great. Okay, a human body is a highly complex system. There are a huge number of tissues we will be studying in this year syllabus. What are the different types of tissues that are present in the humans? Okay, they are made up of the different types of cells. Okay, so this is all the kind of diversity that we are seeing in the cells. Okay, anything you want to ask? Okay, I think we have roughly 5 minutes remaining, and then maybe this is a small topic, that is how small are the cells. I think when we are studying about the microscope, some basic things we have discussed about the dimensions and all. When we speak about an atom, we are talking about lower than the nanometer scale, almost close to nanometer scale. Okay. When we are looking at the biomolecules like the lipids, proteins, okay, some large viruses like flu virus, okay, almost coming to 100 nanometers. But when you look at the cells, a typical bacterial cell, what is the size? Micrometer, 1 micrometer. Okay. So under this light microscope, somewhere around 1 micrometer, a little bit less than that, we will be able to see. Okay. The frog egg can be seen with the naked eye. Okay. But if you want to really look deeper into the details, then we would require a light microscope. Similarly, when we look at animal cells and plant cells, their size will be somewhere between 10 micrometers and 100 micrometers. Okay. And some of the single cells that we can, easily see and is huge. Chicken egg, for example. The chicken egg is made up of how many cells? Can anyone guess? How many cells are there in a chicken egg? It is just one cell. The chicken egg is just one cell, and it is easily visible to the human eye. Okay. So this is a general idea. Okay. How small a single cell would be and how large a single cell can be. The smallest cell are like bacteria, mycoplasma. Okay. Maybe sometime your Olympiad kind of exams may come. Mycoplasma, smallest bacteria. Largest cell is the ostrich egg. The longest cells are the nerve cells. Okay. So this particular we are able to compare the different size dimensions of the cell. Any doubts you have in this one? Okay. So I will take the attendance. Okay. Thank you all.
Thank you, sir.
Thank you, sir.
Thanks.
Sir,
Thank you, sir.
Thank you.